right, well, hello. Today in our little lesson, we're going to be taking a look at how some of the things that we've learned about in the last couple lessons, that idea of genetics, doing Punnett squares, all that, and then we learned about the stages of meiosis and prophase, metaphase, anaphase, telophase times 2. We're going to take a look in this video about how those things actually directly relate to one another. So, and furthermore, I guess, how do we get variation out of that simple process of meiosis? Remember, one of the hallmarks of mitosis was that we got cells that were identical to the parents. In meiosis, we definitely do not get cells that are identical. And there's some pretty important implications, like what we see in this picture right here. This is a picture of one of the largest single families in the world. They're from the UK. I don't know if you can see them, but mom is right here, and then dad is kind of over in this range right there. But somehow, brave souls that they are, have managed to have 16 children. They don't even look that old. They must have started young, I guess. I don't know. But here they are. They're all labeled and numbered. The oldest son up at the top up here is labeled number 1, and they're kind of numbered by their age. So you can see they just had a couple of brand new babies too. It's amazing to me. I stopped after 1. I don't know how I could handle even 2, let alone 16. What I think is really interesting about this, though, is that based on the processes of meiosis, formations of eggs and sperm, these two folks had 16 unique children. Take a look at that picture, all those different faces. There isn't 2 of them that are really all that alike. And neither one of them, or none of them I should say, really look like mom or dad. So how is it possible that that simple process of meiosis, formation of eggs and sperm, can result in this much variation, even within a single family? We're not even talking across a species, but even with the single same mom and dad. My favorite, by the way, is number 8. Look at her over on the right. She looks like you probably feel having to watch this lesson today, right? All right, so just to review. A lot of this is going to feel like review, but we're trying to build up that idea of how is it that meiosis and genetics are directly related. So in the past, we looked at genetics problems like, how do we set up a dihybrid Punnett square with big A, little a, big B, and we talked about that process, that we would take all the different possibilities that you could get, like the first of each pair was big A, big B, the outside was going to be big A, little b, the inside was going to be little a, big B, and then the last pair of each was going to be little a, little b, that we could have these 4 different possibilities. So when we're setting up our big Punnett square, we'd have to have the 4 columns across the top, 4 rows on the side, and this was just a way of dealing with it. The key question, though, of course, comes in this thing over here, which is this idea right here. How is what we're doing here, this big A, big B, little b, actually relate to the process of meiosis? So we've kind of had these 2 separate ideas. Now in this particular video, we're going to try to bring those genetics, those Punnett square ideas, together with our ideas about what meiosis is. So let's take a look at some of these stages. So when we're talking about meiosis, remember it's prophase, metaphase, anaphase, telophase, time 2. Most of this video is going to deal on meiosis 1, because that's really where the key differences happen. I'm not going to talk about meiosis 2 much at all, but we're going to talk about the prophase, metaphase stuff. So what happens in prophase 1? Key idea here is that we've got diploid cells, right? If you take a look at our cells here, we've got 4 chromosomes, 2 big long chromosomes that are kind of bluish in color, and then 2 little short chromosomes that are orangish, reddish in color. Remember, in humans, we would have 23 pairs or 46 total chromosomes running around, much easier with just 4. Each of our chromosomes here is starting off already duplicated. They've got 2 chromatids each. That's why they have that X form. So a key thing that happens in prophase 1 real early on, other than the chromosomes condensing, is that we get formation of tetrads. Now remember what a tetrad is that a tetrad is when somehow, and again, a process we don't really entirely understand, get that guy out of the way, that these guys find one another. So that these pairs of chromosomes somehow match up. We think it has something to do with spindle fibers, but it really even happens before the spindle fibers themselves appear. So it's still kind of a mysterious process. But we get tetrads. Homologous chromosomes find one another. And then as we move on to the next phase, which of course is metaphase, these homologous chromosomes now pair up and then line up. But what's key here, again, and you remember this, is that now the pairs line up. In mitosis, it's single file. In meiosis, it's pairs of chromosomes that are lining up down the middle. So you can see I have some kind of some sample ones kind of lined up here. And then we will also see, for example, formation of the spindle fibers will start to kind of appear this direction, kind of moving out. Same thing on the other side. Sorry, I'm looking at my iPad as I'm doing this. And that those things will start to kind of come out from the edge and collect up. So that's the big key idea here is in metaphase. And you can see in my top tetrad, I've got the little a on the left and the big A on the right. My bottom tetrad, I have big B on the left and little b on the right. could have done it the other way, and we'll actually talk about that here in a little bit. So what happens next? Anaphase, of course, the spindle fibers separate, and now you can see that you've got the chromosomes pulled apart from their partners. And so here, now the way I have it pulled apart, just the way the spindle fibers attached, I've got big A and big B on the left, and little a and little b over there on the right. In the next phase, which of course is telophase, you're going to get that cell formation, right? That cleavage is going to happen. You're going to get that new cell membrane formed down the center, and now we've got the kind of the beginnings of two new cells. And if you take a look at what I've got written on here, is that over on the right, weird, those would be our two gametes that would form from these two cells. The cell on the left would be big A, big B, and the cell on the right would be little a, little b. And those would eventually make eggs and sperm. That would be big A, big B, or little a, little b. But if you remember from that earlier part, that we should actually have 4 different possibilities. And I've kind of already alluded to where those other 2 possibilities come from, because right here we only have 2. And that's the key fact that we see on the next slide. That yes, there was one way that we lined up the chromosomes and separated them in that first bit, but there was a different possibility. And it's really just a matter of chance. Whether this chromosome over here, this little a chromosome, does it get lined up and pulled that direction, or does it get lined up and pulled this direction? It really is just completely chance which spindle fiber attaches from the left or from the right. And it looks in the picture that I have here that it's on the left, but really they're all just mangled up on top of one another. So right now, let's take a look at the second possibility. This is actually independent assortment. That if this little a goes this direction, versus if this little a goes this direction, completely independent of what happens with the bees. This one could go here, or this one could go here. And this is actually how we get our 4 possibilities. So let's take a look at the outcomes if we move them a little different direction. So here you can see anaphase and telophase. Now I moved it differently, and this time I put the little a over to the left instead of the big A over to the left. And so now we have 2 different possibilities. From this one, we finally get to telophase. Of course, in telophase, we're going to get, our new cell membrane is going to form down the middle. It's going to go, right down the middle that direction. Not as pretty a line as my other one. But now when we take a look at our new gametes, we get those other ones that we were looking for. There's little a and little b. Again, this is independent assortment. It doesn't matter whether the little a chromosome goes with the big B chromosome or the big A goes with the big B chromosome, and vice versa. And now we have all 4 of our different possibilities. Keep in mind, going along with this is independent assortment of the sex-linked chromosomes as well. So we've got segregation, right? Big A goes that way, little a goes that way. We've got independent assortment, that the big A pair is different than how the little a pair is lined up, and I guess based on how the tetrads line up and attach. But we also get the same thing going on with the sex-linked chromosomes, that those could be our X chromosomes, for example. Remember, our Y chromosome is that little stunted one. See how much shorter it is? Aww. So cute compared to the X chromosomes, just a little tiny thing. But if we had genes up at the top, you can now see the relationship between, if there was a gene up here for, say, color blindness, that there really isn't a matching section on that Y chromosome. And that has to do along with this idea of variation. So now you could have the big X going with the A one side, or the X and little a going the other side. By the way, this term down here, autosomes. Autosomes are the chromosomes that you have that are not sex chromosomes. So you have 22 pairs of autosomes and 1 pair of sex chromosomes. That's our total of 23 pairs. So going along that last little bit, remember that meiosis 2 is simply kind of like a mitosis division, that we get the kind of the same ideas of what's happening in the sense of segregation, things kind of separating, going one way or the other. So if we take a look at, let's say this was the cell that I ended up with as a result of meiosis 2, we get here is basically metaphase of meiosis 2, and then we would do the anaphase, right? This one would go that way, and this one would go that way. Anaphase would go for this one as well. I know it looks really funny when I do it this way, but too bad. Same thing over here, anaphase, these guys would move away, anaphase, these guys would move away, there we go. And now we actually have the possibility of 4 total cells that are being created. Again, here's our new, going right across the middle. There's our 4 gametes. So if you take a look at this, in the top corner up here, we've got one gamete that's little a, little b, or sorry, little a, big B. And over in this top right corner, we have a gamete that's big A, little b. Bottom is the same. I know it sounds kind of weird. How do we get 4 different gametes? Remember, there was that whole other version of the cells. The mitosis part of this, meiosis 2, really produces 2 kind of identical gametes in the very end. But each one of these, the important thing to realize now is that we have 4 gametes that are haploid cells. They each only have 1 chromosome from each pair, not the 4 that we saw. And these are now single-celled chromosomes. So let's go back to that original picture. How is it possible that all these brothers and sisters can look so different? So if we think back, we had several different ways, 1, 2, 3, 4 different ways, that we could have independently assorted those chromosomes into eggs and sperm. So if we just look at 2 chromosomes, there's 4 pairs, or 4 possibilities. 1, 2, 3, 4. And each one of these could end up being the egg or the sperm that ended up making an offspring. If we looked at 3 chromosomes, when I didn't do that here, I only did 2, it's actually 8 possibilities. 2 to the exponent of the number of chromosomes. And that's kind of a big rule here, because if there's in us 23 different chromosomes, that means there are 2 to the 23rd, 2 to the 23rd different eggs, or 2 to the 23rd different sperm that could be produced by either parent. That's over 8 million different eggs, or 8 million different sperms, just based on independent assortment alone in the process of meiosis. 8 million. Well, when we start combining those together, there's 8 million different eggs from mom, 8 million different sperm from dad. That means there's actually this many different possible offspring. How many is that? Well, here's million, here's billion, 70 trillion possible children could have been produced from your parents. Think about how lucky you are that you're the one that got born, right? 70 different possible ones, and you're the one that's here today. It's amazing. 23 different pairs of chromosomes can result in that many possible offspring, just based on independent assortment. That's the only thing. Independent assortment is what makes this many different possible offspring. So what I want you to take away from this, big ideas. Punnett squares, the way we set them up, are really representing chromosomes. So when we do big A, little a, that's a chromosome with a big A on it or a chromosome with a little a on it. That when we're setting up dihybrid crosses, we're still doing the same thing. We're just looking at 2 chromosomes at a time. So here we've got our different possibilities, right? Big A, big B, little B, little a, little big B, little b, whatever the case may be. I think I said that totally wrong. The key events that we need to take out of this don't happen in meiosis 2. They actually happen in meiosis 1, especially in prophase when we get the formation of tetrads, and in metaphase when we get those tetrads lining up. Because segregation is actually occurring in anaphase. Anaphase 1 is when one chromosome goes one way, one chromosome goes the other way. Independent assortment is simply based on which way in metaphase those tetrads lined up. Did it line up this way? Did it line up that way? And that's what actually creates the different assortments of the different chromosomes. And then finally, the number of different assortments that you can get is 2 to the power of, to the exponent of, the number of chromosomes that you have. So in fruit flies, it's only 2 to the 8th power. In humans, 2 to the 23rd power. It's quite a lot of different possibilities that you can have. And a final thought. There's you, right? The Michael Phelps sperm. I love that cartoon. But there's 2 to the 23rd other possibilities of sperm. Not that there's that many sperm that's made, but just that many different possible sperm that could be made. What would you have been like if one of the other ones had made it first? If you have a brother or sister, you could look at them. Anyway, if you're looking for a 4, there's an idea at the bottom. Find out something about linked genes. It's something I didn't talk about in here, but it's definitely related to this ideas. And then how would linked genes be affected by the process of meiosis in terms of segregation, independent assortment, and the numbers of possible offsprings? And then what would we see in terms of phenotype and genotype ratios? So there you go. Some big ideas. I hope you're feeling a lot luckier to be here today. See you in class tomorrow.